In this lab, we are going to actually cover exercise 9 and 10. 9 is mainly focused on the female reproductive system, and exercise 10 is male. So let's start with the overview of reproductive system. Let's first look at what does the reproductive system do. Right? The major function of the reproductive system is to produce sex hormone, but also produce gametes, the sex cells, and not only produce that, but for maturation as well as for release of the gametes. Right? The gonads is the primary the organ that's doing this. So in the female, the gonad is ovary, and the gametes is the secondary oocytes. There are actually different types of oocytes, but only the secondary oocyte is the gamete. We're going to go through all the different types of oocyte later. And in the male, the gonad is testis, and gametes, the sex cell, is sperm. Right? So when a sperm fertilizes oocyte, secondary oocyte, this is called fertilization. They fuse together, the nucleus fuse together, right? So this generates, the fertilization generates a zygote, and zygote will go on to develop into offspring. So those are the major gonads, right? But the reproductive system there's additional accessory reproductive organs in both male and female. That basically what they do is to carry, right? They include ducts to carry gametes towards the site of fertilization or to the outside of the body. So that's what the other reproductive system organ do. We'll first look at how this process of generating gametes is called gametogenesis, right? So genesis meaning to create, gameto meaning creating gametes. Right? In the female, we already talked about, right? The gametes is the secondary oocyte, so this process is also called oogenesis. In the male, the gametes is sperms, so therefore the male gametogenesis is also called spermatogenesis. And let's have a look what really happened in this process, right? So it's basically we probably, if you remember in MP1, we talk about, the normal human cells are called somatic cells, right? They are diploid, right? They have 2 copies of the same chromosomes. So each one of us, right, each one of those somatic cells have 23 pairs, because for each chromosome they have one from mom, one from dad, so they have a copy, a pair of the chromosomes. And then out of those chromosomes, 22 of them are called autochromosomes, right? And then one pair is called sex chromosomes. So that's X and Y is the sex chromosomes. And then when we, for each pair, like the 23 pairs, so if you have a pair of 21 here, the 2 copies of the 21 chromosomes, they are also called homologous chromosomes. Okay. For the gametes, however, for each gamete, it only contains 23 chromosomes, not 23 pairs. So each chromosome, each pair of the homologous chromosomes, you only have one of them end up in gametes, right? So gametes do not have pairs. So that's a critical concept. Well, how do we know whether it's a diploid cell, right, or it's a haploid cells? Well, to see whether they have homologous chromosomes. If there's no homologous chromosomes, right, So you only have 23, not the 46, which has 23 pairs of homologous chromosomes, and that's haploid, right? So we'll see an example of this. Let's first look at some basic terminology, right? We talk about chromosome. What is a chromosome? It's basically the DNA, right? Condensed, supercoiled, and into a chromosome that's linked by a centrosome. And then we'll have a pair of homologous chromosomes, right? One from mom, one from dad. So they look like very similar, and there are two of them. So that's usually in non-dividing cells. That's a single chromosome, that's a pair of chromosomes. In the dividing cells, right before the cells divide, so the DNA has to be replicated, right? So this is what happens. You have replicated DNA, but still connected by one centromere, right? But then there are two sister chromatids. So this is still called a single chromosome. And then the pair of homologous chromosomes, then you have still one of them. You get this one from mom, but then it has been replicated, but it's still a chromosome that made out of two sister chromatids. So it's called tetrad because it's actually there's four times of the genetic material here than the normal single non-divided chromosome, right? So it's but then in the meiosis, they have to line up, right? Before the division, they have to line up, but then crossover can happen here. But this only happens in meiosis. So that means crossover, meaning part of the chromosomes are swapped, right? So you end up having none of those single chromosomes is exactly the same as the chromosome you inherit from your mom. It's another way to generate more genetic diversification. And so this is actually, but then in the mitosis, no crossover happens. So you basically only have the split of the sister chromosomes, right? So then the two component, what happens is identical, like offspring. The daughter cells are identical, right? Then the cells you start with this, you end up with, this. That's what happens in mitosis. But in meiosis, that's the process that generates gametes. There's two rounds of division happening. And before the first round happens, crossover happens before the first round of division. Since AMP1 have covered mitosis, we only focus on meiosis in this part, in this chapter. So meiosis, like I said, can be divided into meiosis 1 and meiosis 2 because there's two rounds of division. First round, you start with, the tetrad, right? But then crossover has already happened, right? You already have swapped the homologous chromosomes for part of the chromosomes, and they're going to line up in the middle of the spindle. But then look at here, the lineup is not single line. Instead, there's two. They line up in two rows. So this is different from what happens in mitosis. And then this is metaphase 1 and then anaphase. So those terminology are similar as the ones in mitosis, right? You have the prophase, metaphase, anaphase, and telophase. So then because they line up in two rows, but they randomly, right, the chromosomes are randomly aligned in one of those two rows. So when anaphase happens, the basically homologous chromosomes got to sort into different daughter cells, randomly, right? So not all the chromosomes you inherit from mom is going to be sorted in one end. Instead, it's randomly, the homologous chromosomes are randomly sorted into 2 daughter cells, right? Now you end up with, let's say, if you started with 3 pairs, like shown here, right? Three pairs of chromosomes. Each of those daughter cells, you only have 3 chromosomes. So that means, are those cells diploid or are they haploid? Well, since they do not have a homologous chromosome to go with them, right, to be in the same in the same cell, so those cells are haploid. And then for meiosis 2, let's just look at one of those daughter cells from meiosis 1, right? Then you have metaphase 2. So then they line up now, this time in the middle, right? There's only one row between the 2 poles. This here you have the separation of the sister chromatid, right? So the daughter cells you end up with then each one of them still haploid, but then you do not have the sister chromatids anymore. The replicated DNA are separated into the 2 daughter cells. The same thing goes for the other daughter cell from the meiosis 1, right? So the end is, if you look at those 4 daughter cells, do you have unique daughter cells, or do you have all the daughter cells have exactly the same DNA material? If you look at all those, right, they actually, none of those are the same. They all have different combination. They inherit some of the chromosomes, right, from your your parents, but then they're swapped, additional recombination happens essentially. So the 4 daughter cells are actually from the meiosis are different. They're different from each other, right? And so that's a big difference for meiosis versus mitosis. Mitosis, you end up for each division, you end up with 2 identical daughter cells, and each one of them is the same as the one you start with. But for meiosis, not only you ended up with haploid cells, not diploid cells, but also they are unique. Each one of them are different from each other. So that's the process of generating gametes. You need to actually draw this for the exercise, to draw this process. And so then I would suggest you to really compare that with mitosis. That's in chapter 4. You learn that in AMP1. Just in case if you forgot about it, compare what you draw in the exercise, second page of the exercise, then to the mitosis, and see. So only by comparison you start to apprehend the difference between them. And you also need to be able to then answer the questions that the last 3 rows in the page 2 of the exercise 9. Ask you questions about what's the result of mitosis, what's the result of meiosis 1, what's the result of meiosis 2. So let's then move on to then look at the female reproductive organs. So female reproductive organs, we said the most important one is the gonad ovary, but then it also has the accessory reproductive system like the uterus here. This is where the embryo and further later fetus develop, right? You have a tube that connecting the, basically pick up the ovulated egg, right, from right next to the ovary, then and then basically connect that to the uterus. That will be called uterine tube. There are different terminology. Fallopian tube is an older term we use. In this textbook, we predominantly use uterine tube. Sometimes you say oviduct. That's another name for that. And then, so the first part of the uterine tube is this finger-like structure, right? So then you can kind of create, there's cilia cells there, create a swirl, then basically can pick up the oocytes that's ovulated, then from right next to the ovary. And so this part, finger-like type of the structure is called fimbriae of the uterine tube, fimbriae, right? And then, so that's the uterus. That's a pretty muscular kind of sac, right? Then the opening of the uterus is called cervix. Cervix of the uterus then is opening to the vagina. And the vagina is going to basically lead to the outside of the body, and that's the orifice of the vagina. Vaginal orifice, that's basically opening of the vagina. You can see anterior to the vagina orifice, you have the external urethral orifice. So this would be the urinary bladder, right? That's the urethral. So those two are right next to each other, the urinary bladder and the uterus. If you have to remember, one way to remember those will be the uterus is more posterior, right? The vagina and uterus are posterior than the urethral and the urinary bladder, position-wise, right? And the uterus is actually sitting on top. Part of it is sitting on top of the urinary bladder. So here is a look, another look of the those female reproductive. Just look at the internal structures, right? So you have the uterus here. This is from posterior view, right? Then the two ovaries on the lateral side. The uterus and the ovary and ovaries are sandwiched between this broad ligament. So then it's keeping that that reproductive system in place. So also sandwiched within that, you have the ureter sandwiched within this broad ligament, right? And also the blood vessels also are sandwiched. So then it's actually secure, right? It's in the pelvic cavity. And so the ovary is also attached to the uterus by this ovarian ligament, right? It's also a ligament. There's no ducts in that, right? So it's not a duct directly connecting the ovary to the uterus. You have the uterine tube, basically pick up the egg right next to the ovary, but then it's still not directly connected to the ovary. The uterine tube is not directly connected to the ovary. It is directly connected to the uterus. So this is a side view, a lateral view, looking at the structure. So you have the ovary right here, right? So you can see the ligament is basically just connective tissue, right? A dense connective tissue connecting the ovary, right, to the broad ligaments right here. So the cross section is right here. And so that's, but if you look at this cross section of the ovary, you'll see a bunch of follicles, different size, right, in it, and some blood vessels. And so that will be called cortex. And internal will be called a medulla. You are familiar with those kind of setup, right? But then on the follicles are all on the different sides of the follicles are all to the in the cortex of the ovary. And let's have another look, a closer look of what kind of the follicles are in the cortex of the ovary. So here you can see the medulla is mainly full of the blood vessels, right? And if you start from the smallest, right? The smallest follicles. Those are called primordial follicles. They're kind of the, you have those follicles ever since you basically develop as a fetus in your mother's womb, right? So every month, some of them are going to start to, develop. And so they become bigger, the ones that to develop called primary follicles. And after a few primary follicles, only one of them going to be able to, every month, there's only one of that going to be able to develop into a mature follicle. So depending on the size, the biggest one is the mature follicle that's ready to be ovulated, right? And then, so if you look at the size, that's the most striking, is the biggest one is about to be erupted, called mature follicle, right? But then you also have secondary, so primary follicle, secondary follicle, right? And then this one is called antron follicle. Antron follicle, then after the eruption, the mature follicle erupt. So this is the oocyte, ovulated secondary oocyte, together with its supportive cells. You have a zone of pellucida, is this transparent kind of zone of protective material, and then corona radiata. Corona meaning because it's like a crown that's surrounding the oocyte. So coronal radiata. So that's the mature, that's the secondary oocyte being ovulated, right? Released from the ovary. And after this happens, the reminiscent of the mature follicle, the surrounding tissue, is going to develop into corpus luteum, and that's going to be continue. This corpus luteum continue to secrete hormones, and mainly progesterone. And then so it's corpus luteum is kind of the reminiscent of the follicles, the mature follicle, right? And eventually the corpus luteum is also going to regress into some kind of whitish color. So that's called, luteum meaning is yellow color, and albicans meaning the white color. So corpus albicans meaning the white reminiscent of the follicle. That's the end of the follicle. So this is kind of the cycle, right? Each follicle, if it develop, is going to go through this cycle. And we are going to have look at some detailed structure that distinguish the primary, secondary, antron follicle, and the mature follicle. So in this slide, I listed all the types of the follicles, right? The primordial follicles are the smallest and underdeveloped. The primary follicles are bigger than that, right? You have the primary oocyte in here, then surrounded by a zona pellucida, this transparent kind of layer, and then granulosa cells, one layer of granulosa cells. And when it grows bigger, it's mainly the granulocytes multiply into multiple layers, right? So you have a thickened zona pellucida. The primary oocyte size didn't really change. And then when you get to the antron follicle, it's mainly because you have this appearance of the antron, which is a space, right, between the granulosa cells and the corona radiata. So you start to see this space that's called antron. So the appearance of this antron, then that's the signature that develop into the antron follicle. Antron follicle then develop further into the mature follicle. You have a expanded space, right, of the antron, right? So the zona pellucida and the follicle cells and the oocyte itself are are still roughly the same size, but then the corona radiata also becomes multiple layers, right? So at this stage, once it gets to the mature follicle, the oocyte here is the secondary oocyte. What's the difference between primary oocyte and secondary oocyte? We're going to mention, go through what happened inside of the cells depending on which stage of the meiosis it is. We're going to focus on that in the lecture. But for this is what the corpus luteum look like. It's yellowish, right? Have some space in the middle, and the corpus albicans is whitish. So that's the last stage of the follicle. You need to be familiar with those. So if I basically have labeled one of those in the diagram, the last diagram in the slides, and you should be able to name that. Okay, name which type of follicle that is, and and be familiar with all those structures. In the exercise, you're supposed to draw them to draw and label the cells, right? The zona pellucida, granulosa cells, and androme 2. So it would be best if you draw that and then compare them, so that you'll have a better understanding of different life stage of a follicle disease, actually, because for each one of the mature follicle, you would have to go through all this stage. So this is really just a kind of life cycle kind of diagram. But because in the ovary they have multiple follicles in different stages, that's why you can actually see all of this, right? Follicles, all those different types of follicles in the ovary, but they represent different stage of a particular, each single one of the follicle. It's just because if you look at the snapshot of the time, then you do see different follicles caught in the different stage of development. So here and this diagram is going to focus on the uterine tube and the uterus. So the uterine tubes are actually can be divided into 4 major parts. The first part is the infundibulum. That's the part with fimbriae. So that's the one, with the funnel shaped, right? And not only is funnel shaped, but it also have fimbrian kind of finger-like structure, kind of swarming around, right? And the second part is from the bottom of the infundibulum, then to about 1/3 of the length of the uterine tube. That's called ampulla. Ampulla is where actually the eggs get fertilized. And then the next part is called the remaining 2/3 of the fibrin tube is called isthmus. And then where basically the uterine tube is connecting right to the uterine, that part is called uterine part. Those are the 4 parts of the uterine tube. If you look at the cross section of the uterus, and uterus is really kind of a pear-shaped organ, right? Upside down. And it has a muscular kind of wall. That's the one we talk about, right? If you look at the cross section, the most inner layer of the uterus is called endometrium, right? Endometrium. Then the muscular layer is called myometrium. Myo meaning muscles, right? And the last, kind of the connective tissue layer is called the perimetrium. So that's the wall. Those 3 layers makes up the wall of the uterus. But if you focus on the endometrium, that's the part you have, growth and shedding in each menstrual cycle, right? So you have the endometrium. There are 2 layers. You have the functional layer and the basal layer. Okay, So that will be the lumen of the uterus, right? Because this is the inner metrium, the one closest to the lumen. On the top, there's epithelium, right? You have in those functional layer and basal layer a lot of uterine glands, okay? And then underneath of the endometrium, you have the myometrium. So this is the histology slide of the uterus. In the last part of the exercise 9, your exercise also asks you to basically summarize, look at APR and your book, and summarize, draw ovary cortex histology slides. So that'll be helpful for you to look for some APR structures. I don't think we have a really good kind of overview cortex of ovary cortex picture in the book, but you could use the resource in the APR to do that.